The two classes of MHC molecule have distinct subunit structures but similar three-dimensional structures. The two classes of MHC molecule differ from each other in their structure and also have different distributions on the cells of the body. Their different structures enable the two classes of MHC molecules to serve distinct functions in antigen presentation, binding peptides from different intracellular sites and activating different subsets of T cells. Despite their differences in subunit structure, however, MHC class 1 and class 2 molecules are closely related in overall structure. In both classes, the two paired protein domains nearest the membrane resemble immunoglobulin domains, whereas the two domains distal to the membrane fold together to create a long cleft or groove, which is the site at which a peptide binds. Purified peptide MHC class 1 and peptide MHC class 2 complexes have been characterized structurally, allowing us to describe in detail both the MHC molecules themselves and the way in which they bind peptide. MHC class 1 molecules consist of two polypeptide chains, a larger alpha chain encoded in the MHC genetic locus, and a smaller, non-covalently associated chain, beta-2 microglobulin, which is not polymorphic and is not encoded in the MHC locus. Only class 1 alpha chain spans the membrane. The complete molecule has 4 domains, 3 formed by the MHC encoded alpha chain and 1 contributed by the beta-2 microglobulin. The alpha-3 and the beta-2 microglobulin chains have a folded structure that closely resembles that of an immunoglobulin domain. The most remarkable feature of the MHC class 1 molecules is the structure of the folded alpha-1 and alpha-2 domains. These two domains form the walls of a cleft on the surface of the molecule. This is the site of peptide binding. They also are sites of polymorphisms that determine T cell antigen recognition. An MHC class 2 molecule consists of non-covalent complex of 2 chains, alpha and beta, both of which span the membrane. The MHC class 2 alpha and beta chains are both encoded in the MHC. The crystallographic structure of the MHC class 2 molecule shows that it is folded very much like the MHC class 1 molecule. The major differences lie at the ends of the peptide binding cleft, which are more open in the MHC class 2 molecules compared with the MHC class 1 molecules. The main consequence of this is that the ends of a peptide bound to an MHC class 1 molecule are substantially buried within the molecule, whereas the ends of peptides bound to MHC class 2 molecules are not. Again, the sites of major polymorphism are located in the peptide binding cleft, which in the case of an MHC class 2 molecule are formed by the alpha-1 and beta-1 domains. And that's the two classes of MHC molecules have distinct subunit structures but similar three-dimensional structures. Have a great day.